Okay, so when we're talking about codons, the genetic code has several different qualities to it. First is that the genetic code is degenerate. And what we mean by this is that most amino acids are going to have more than one codon coding for them. As we saw in the previous slide, there's 64 different possible combinations, but only 20 amino acids. And so what that means is that most of these amino acids are just going to have more than one triplet code that will code for that same thing. The second is that the genetic code is unambiguous. If you have a specific sequence, it is only going to mean that specific sequence and nothing else. And so each triplet code is only going to have one meaning. If it didn't, things would start to get very confusing and it would make no sense in writing a gene down because who knows what you're going to get with it. And so each code absolutely going to be unambiguous. The next thing is that the genetic code has start and stop signals. There are very specific sequences that the organisms can use to start a transcript or end a transcript. When we're reading these codes, there is no comma to them, there's no pause, it just starts until it gets to the end. So it's commaless. Also, there isn't any overlapping happening at the same time, and I'm going to talk more about that in just a minute. And the last thing is that these codons are going to be almost nearly universal. So if we look at the codon chart here, you can see a couple of the things that we just talked about. First of all is the degeneracy. So you can see most of these amino acids are going to have more than one codon that will be coding from them. The way that you read it is you look at the first position of 3, and it's over on the left here. The second position is here, the third position is here. So if you have something coding for, say, GAU, that means you're going to be coding for aspartate. Okay? GAC, also going to code for aspartate. So we see right here the degeneracy of the genetic code. We can also see the start and stop signals. AUG is methionine, but it also is the initiation code or start code. We have 3 termination signals, UAA, UAG, UGA. These all mean stop. Only 2 amino acids have only one codon for them, and that is methionine as well as tryptophan. Everything else is going to have at least 2 codons that will be coding for it. And this leads us to our initial hypothesis. It's called the Wobble hypothesis. And what happens is that the initial 2 nucleotides of those triplet codes is actually more critical than the third member in terms of attracting the right tRNA. And that becomes more important. Again, next chapter we'll talk about that. The reason for this, probably due to the fact that hydrogen bonding at that third position just less spatially constrained. And so what it means is that if you have some similar amino acids, let's see here, so looking at the Wobble hypothesis, again going back, if you have a CUG or a CUA, CUC, CUU, these are all going to code for a leucine. And similarly here, proline, CCU, CCC, CCA, CCG, they're all coding for a proline. And the only difference is, again, in that third base of that codon. Another thing that we see is that chemically similar amino acids are going to share 1 or maybe 2 of those middle bases, the first base or 2, in the triplets that are encoding them. So, for example, if you have an AAA or an AAG, this shows you not only the Wobble hypothesis, but also this is a code for lysine, which gives you a positive charge. If you look at AGA or AGG, very similar, just changing that central base for that codon, you're now going to be coding for an arginine. This is a very chemically similar amino acid. Both of these are going to have a positive charge, net positive charge. And when we get things like this happening, what's happening is that it helps to buffer this potential effect of mutations on protein functions. Say you were getting a mutation and you change this base from an A to a G. Well, you're changing that particular amino acid then from a lysine to an arginine. This isn't actually a huge deal because they are similarly charged or similarly sized, and so it's just going to buffer or help to negate a little bit some of these potential effect on mutations on that protein function. So maybe at least would still be somewhat functional. And like I mentioned before, only methionine, only tryptophan are the ones that have a single codon assigned to them. So I also mentioned that the genetic code is universal, and I put a little asterisk by this because there are just a couple of exceptions that we'll talk about. But for the most part, that codon chart that I showed you, that is the same. It doesn't matter if you're talking about a bacteria, doesn't matter if you're talking about a human, doesn't matter if you're talking about your cat or your dog. Okay? That codon chart would actually be the same in all of these organisms. And so this is not only strong evidence that all living things are going to share a common evolutionary heritage, it also is a pretty cool way in biotechnology that you can transfer genes from one organism to another. And so you can take the human gene for insulin, you can put it into bacteria, and those bacteria will grow it up for us. We can purify it, and then humans can use this for medicine. And there are many, many examples of this. So here's a couple of the exceptions to the universal code. Most of them center around mitochondria, some paramecium, tetrahymena, which I believe is a protist, and a few other things. But as you can see, for the most part, there's just very few exceptions to that universal code that we talked about on the previous page. Lastly, you can have different initiation points which would create overlapping genes, but within a particular sequence. None of those codons are going to overlap with one another. Those codons are all read one after another after another. Sometimes when that genetic material needs to be packaged very, very tightly, you can have overlapping genes, such as the case with some viruses, and we'll call these overlapping genes an open reading frame, or ORF. And what you can see here is they all have an AUG for their start. Here's an AUG here, and it's going to start to make this particular transcript. This AUG here is a start, and it's going to make a completely different transcript. Now this is out of frame from this, but it doesn't matter because within its own frame, it's in frame. And you can see here an example what you might see with a typical virus. You can see here A prime, B is kind of overlapping, K is overlapping here, C, D, and E are overlapping. And as long as within that frame that you stay in frame of the three bases, everything will be fine.